Hi, my name is Julian and I am in the last year of my PhD at Uppsala University in the Berger lab. And I will present to you today a chapter of my thesis in which we were looking for experimental evidence for the effects of sexual selection on condition-dependent mutation rates. So, since quite a while now, we have understood that mutation rates are variable, and they vary between species according to genome size and population size. And if you look in this figure, you can see in the bottom left corner, there are small microorganisms that have relatively small genomes and live in huge populations. While in the top right corner, you see mammals which have large genomes and relatively small populations. And so even though on the x-axis you have genome size, what is driving this relationship is primarily the population size, because the strength of selection is directly proportional to the size of the population. So in a smaller population, a given mutation has much less negative effects on an individual than it has in a huge population. And that leads to the fact that in animals that have smaller population sizes, there's less pressure to have super high fidelity of DNA repair in the germline. But the other level on which the mutation rates are variable are within species, and that is between individuals of different condition. And if you look at this figure here, this is from an experiment where they had populations of a seed beetle that they kept in a benign environment and populations that they kept in a stressfully hot environment. And what they found is that the individuals that were in the stressful environment, they also had a higher mutation rate. And some of you might maybe be wondering now what exactly this condition is that I'm talking about. And so the condition is a term from evolutionary theory which refers to the pool of resources from which any given individual can take to allocate to any of its body functions. So you understand that this is an extremely broad and an extremely vague definition, but it generally means something along the lines of the quality of the phenotype of this individual. And as such, it can be affected by a variety of factors. Some of them are environmental, like for example, the availability of food, or as in the previous example, the temperature, and so on. Then secondly, we have genetics, which refers to the genotype of an individual. So an individual which, for example, has a genetic disease has a phenotype of lower quality than an individual that has a healthy genome. And last but not least, we have the current health status of the individual that can, for example, refer to how high its parasite load is. And this condition is quite important in evolutionary theory. It has, for example, been invoked to solve the so-called leg paradox. And so the leg paradox is basically the question of, if we have strong directional selection on any given trait, should that not over time deplete the genetic variation for this particular trait in a given population? And to give you an example of this, we have these American sage grouse here on the right. And in this species, the males and females, they meet in a so-called lek every mating season. So it's basically a mating arena. And all the males display their tail feathers here. And the female chooses the one male which has the longest, most beautiful tail feathers to mate. So what we would expect is that over time, all of the males in this species should show the most beautiful display of tail feathers. However, if we go out in nature, there is still quite some variation for this trait. How can this be? And so a solution to this question has been proposed by Rowe and Houle, and that is the so-called genic capture hypothesis. And what they thought is that if the expression of this trait, tail feathers in this example, depends on the condition of the individual, that could support the amount of variation that we see in nature, because as we have learned on the previous slide, condition depends on the entire genome. So any mutation that has a deleterious effect and occurs at any point in the genome can affect the expression of the trait. So this increases the target for deleterious mutations to lead to variation in this trait from just the few genes which are probably relevant for this trait to pretty much the entire genome. So, but mathematical estimates have shown that to get to this variation that we see in nature, we would still need mutation rates that are quite a bit higher than mutation rates that we can directly measure. So what we thought in our lab is that maybe sexual selection can directly have an impact on germline maintenance and therefore the mutation rate. And we came up with two different options how this can go down, which I will try to explain to you using this classical Y model for trade-offs, which of course is a very simplified approach, but for now it should be sufficient. So a first case how we could see this play out is that if there is some flexibility in the allocation to the different functions as reproduction, as in the investment into sexual selection, or germline maintenance equaling to the mutation rate in the end, sexual selection could select for males which are able to invest more into reproduction and neglect germline maintenance or other functions of their body. And so over time, sexual selection would lead to males that can invest increasingly more into reproduction at the cost of other functions, which would lead to an increase in mutation rate under sexual selection over time. And the other option that we could imagine is that if there's no flexibility in the outcome here, and the only way that the investment into reproduction can be increased is through an increase of the influx of resources into the system, or in other words, the condition of the individual, then we would end up with a lower mutation rate after some time with sexual selection. And to test these two ideas, we designed an experiment using Callosobruchus maculatus seed beetles. And so this experimental design has three important parts. It starts off with 29 generation of experimental evolution. Then we have like an immediate social sexual treatment, which basically reflects investment into sexual selection in this exact moment. And we have an assessment of the mutation rate in the end, which is not very important for how exactly we did this for the appreciation of the results that we found. So first, the experimental evolution regimes that we applied for 29 generations in these populations are the following. In a first set of replicate population, we allowed for natural selection as well as sexual selection. And this is the natural mating system of this particular species. And natural selection in the species occurs mainly through fecundity selection on females. So each female lays a number of eggs and they contribute proportionally to this number to the next generation. And sexual selection in this species is acting on males mainly, and it is pre-copulatory competition, but also post-copulatory competition in the form of, for example, sperm competition. And so in a second set of populations, we completely remove the opportunity for sexual selection to act by just assigning one male at random to every female. But then the females can still lay as many eggs as they want to contribute proportionally to the next generation. So if we compare these two sets of populations, we get the direct effect of sexual selection. And then in a third set of population, we have removed the opportunity for fecundity selection on females. And by just taking the same amount of offspring from every females, but the males can still compete for the females. So comparing this to the first set of populations gives us the effect of natural selection. And then after the experimental evolution, we had another treatment which we applied just in the 24 hours before the assessment of the mutation rate on these males that we assessed the mutation rate on. And so half of them were allowed to compete with each other and mate with females and basically just invest into sexual competition, while the other half was kept isolated and virgin in small Eppendorfs. And then we come to the results that we found. And so on the y-axis here, you have the mutation load, which is basically proportional to the mutation rate. And if we just compare the populations that evolved under natural and sexual selection with those that evolved under natural selection, we see that the effect of sexual selection seems to lower the mutation rate in these populations. And this would point towards this option where sexual selection acts mainly on the condition. But if we look at the populations where we only applied sexual selection and no natural selection, we can see that there is a plastic increase in mutation rate if they were exposed to this social sexual treatment right before we assessed the mutation rate. So we can still see the overall positive effect of the experimental evolution treatment, but we see that in this population, there seems to be a trade-off with this investment into this competition and the mutation rate. So what we can say as a summary is that we find a lower mutation load in the populations that evolve under sexual selection, which is in line with selection for better condition. However, in the populations that evolved under sexual selection and in the absence of natural selection, we find a plastic increase of mutation rate if we expose them to competition and mating, which supports this direct trade-off between the two. So in conclusion, we can say that strong sexual selection can, in certain cases, lead to an increase of mutation rate. And these findings can help us explain and have implications for the maintenance of genetic variation in traits under strong directional selection, but also the sexual dimorphism that we find in many species for mutation rates. And last but not least, the efficacy of purifying selection in populations of sexually reproducing species. And with this, I would thank all of you for listening, and my boss and our lab assistant for their help during this experiment. Thank you.